In this video, we're going to talk all about prokaryotic cells or bacterial cells. So we're going to go ahead and have a go at labeling all of the key structures in a prokaryotic cell, and then we're going to go through and look at the structure and function of those structures as well. So here is a typical prokaryotic cell. Now, in terms of a prokaryotic cell, what that really means is it has no nucleus. In fact, it has no membrane-bound organelles. So not only does it have no nucleus, but it has no mitochondria, it has no Golgi apparatus, no Ruffia, no Smoothia, no membrane-bound organelles at all. Let's label. So if we start over here, I is pointing to the inside layer of 3. So this is going to be the cell surface membrane or the plasma membrane. H is pointing to the second layer of 3. So this is going to be the cell wall, which, by the way, is not made of cellulose in a prokaryotic cell. It's actually made of something called murin, or an alternative, we can call it peptidoglycan. So it's a different molecule. It is not made of cellulose, and that's one of the differences that you could give between like a prokaryotic cell and a eukaryotic plant cell. The outer layer is the capsule, or the slime capsule, as we call it. Down here we have a flagellum. Now this bacterial cell has one flagellum. Some bacterial cells have many flagella, some don't have any. So bear that in mind. E is a ribosome. Looks a little bit big, that, to be a ribosome, doesn't it? But it's got to be a ribosome, there's nothing else it can be. Remember, this is a smaller 70S ribosome. D is cytoplasm. C is, we can call it the nucleoid, if we want to, or we can just call it circular DNA, okay? But it's circular DNA, it's not inside a nucleus, it's loose in the cytoplasm, but a word that we can use to describe that main bulk of DNA in the prokaryotic cell is nucleoid. B is a plasmid, so a smaller circular loop of DNA, and A, they are pili. Now, what I'm going to do is just underline the structures that not all prokaryotes have. So not all prokaryotes have pili. So it's not a good idea to suggest that as a difference between a prokaryotic and a eukaryotic cell. It's not a good idea to say prokaryotic cells have pili, eukaryotic cells don't, because what I'm saying is not all prokaryotes have them anyway, okay? Another one, the slime capsule. Not all prokaryotes have a slime capsule. Not all prokaryotes have flagellum, and not all prokaryotes have plasmids. So I wouldn't be naming these structures as structures found in a prokaryote, not in a eukaryote, when not all prokaryotes have them. So just be a little bit careful there. Let's go through now and think about the kind of structure function. So starting with the circular DNA, or as we said before, we can call it the nucleoid. The nucleoid. I'm going to get my highlighter, I think. So it codes for proteins. That's a really simple way to say it. But I think at A level, we should be able to say the order of bases determines the order of amino acids in a polypeptide. Remember, 3 bases codes for 1 amino acid, and the sequence that the bases are in will determine the sequence of the amino acids in the polypeptide, which makes the protein. Plasmids, remember, not all prokaryotes have them, but they are small, circular, or loops of DNA. They're independent of the main DNA. So the main DNA is obviously the nucleoid, the main genome. But the plasmids are smaller circular loops of DNA not associated with the nucleoid. They often carry genes that provide a selective advantage. So genes for antibiotic resistance will be found on the plasmids. But remember, not all prokaryotic cells have them. The capsule, or the slime capsule, again, not all prokaryotic cells have them, but in terms of function, they protect against desiccation, which just means drying out. They shield the cell from phagocytosis. So the phagocytes, which are a type of white blood cell that form your immune system, having a slime capsule kind of covers the antigens a little bit. It's like a little cloak around the bacterial cell, and it can protect them from phagocytes or phagocytosis. Also helps the bacteria to adhere or stick to other surfaces because it's like a gelatinous, sticky slime-like material. The cell wall Remember, not made of cellulose, it's made of murin or peptidoglycan. It provides structural support just like it does in a plant cell. It helps maintain the cell's turgidity. It can withstand turgor pressure. So if the bacterial cell does fill up with water, that cell wall is going to provide structural support, just like it does with a plant cell. Primarily composed of peptidoglycan, but remember, we can call peptidoglycan murin. If that's what you've learned, that's absolutely fine. Let's move on to the flagella. Again, not all bacterial cells have flagella, but they are long whip-like appendages. They provide motility, so it makes the prokaryotic cell able to move. For example, they can show positive chemotaxis, which is something you'll learn about in year 13. So they can move towards positive, a favorable environment, or towards a chemical, or they can move away from harmful ones, which would be negative chemotaxis. Animal cells, think about a sperm cell. That is one type of animal cell that also has a flagellum, and obviously that flagellum enables the sperm cell to move or swim towards the egg cell for fertilization. Pili, again, not all prokaryotes have them, but they are hair-like structures. They're on the surface of the prokaryotic cell. Their functions include conjugation, which we don't really learn about anymore in A-level biology, but they do connect bacterial cells together, and you can get the transfer of genetic material between bacterial cells. It's horizontal gene transfer. So for example, a plasmid from one bacterial cell can be passed to another bacterial cell using those pili, or we'd call them conjugation tubes. Also used in adhesion, so pili can help the bacteria attach to surfaces or to host tissues. Now, let me just go back to my pen for a second. Other things just to note, I think, underlining again, just because this has caused some confusion in exam papers, not all prokaryotes have plasmids, not all prokaryotes have that slime capsule, not all prokaryotes have flagella, not all prokaryotes have pili. So if we are focusing on differences, we should be focusing on differences in the cell wall of, say, a prokaryotic cell and a eukaryotic plant cell, differences in the circular DNA or the nucleoid in a prokaryotic versus a eukaryotic, or differences in the ribosomes, which I haven't put on my list because I'm confident we know they are for protein synthesis. But remember, in a prokaryotic cell, there is only 70S ribosomes. Whereas in a eukaryotic cell, you can have 70S ribosomes in the chloroplast or in the mitochondria, but you also have 80S in the cytoplasm and on the rough endoplasmic reticulum. Okay, let's have a look at the final slide. So describe the differences between these two types of cell. So prokaryotic versus eukaryotic. If we go for prokaryotic first, there's no nucleus, there's no mitochondria. In fact, we could bracket them together, and for 1 mark in an exam, you can say there are no membrane-bound organelles. Now, obviously, if you were doing this in an exam, you would have to give the difference. So you would then go on to say, whereas eukaryotic cells do have a nucleus, they do have mitochondria, or they do have membrane-bound organelles, which include the nucleus and the mitochondria. Prokaryotic cells only have 70S ribosomes. Now, that word only is important because eukaryotic cells have 80S and 70S ribosomes. So what we can't say is prokaryotic have 70S and eukaryotic have 80S. It's not quite right, is it? Prokaryotic only have 70S. Eukaryotic do have 70S, for example, in the mitochondria or in the chloroplast if it's a plant cell. They also have 80S, obviously in the cytoplasm and on the rough endoplasmic reticulum. Moving on to the DNA, prokaryotes have a nucleoid, or they have circular DNA. It's also not associated with histone proteins. And the other thing we might know is it doesn't contain introns, prokaryotic DNA. Whereas eukaryotic cells, they have linear chromosomes. And these linear chromosomes are associated with histone proteins because the DNA wraps around histone proteins. And there's also introns in eukaryotic DNA, which are the non-coding sections of DNA. Now, again, you have to be careful with this because there is some circular DNA in a eukaryotic cell. In the mitochondria, there's circular DNA. In the chloroplast, there's circular DNA. But the prokaryotic cells only have circular DNA. Yeah, they only have circular DNA. They do not have any linear chromosomes or any linear DNA. They only have circular DNA, or we can say that they have a nucleoid. You'll notice that I'm not talking about flagella. I'm not talking about pili. I'm not talking about a capsule. I'm not talking about plasmids. It often will not be accepted on the mark scheme because not all prokaryotes have them. So I'm focusing on specific and clear differences. The other one that I've just remembered that you could have is for a prokaryotic cell, the cell wall is made of murin or peptidoglycan. Whereas for a eukaryotic cell, don't just say they don't have cell walls, because plant cells are eukaryotic cells and they do. So we could say plant cells, or if they do have a cell wall, it's made of cellulose. Okay, now I've not written this very nicely. Obviously, when you're doing this in an exam, you match up the differences. So you structure it better than me. You say prokaryotes have a cell wall made out of murin, whereas eukaryotic plant cells have a cell wall made out of cellulose. Prokaryotic cells only have 70S ribosomes, whereas in eukaryotic cells, they have 80S as well as 70S ribosomes. Okay, prokaryotic cells have a nucleoid, or they only have circular DNA, whereas in eukaryotic cells, we obviously have linear DNA in the form of chromosomes in the nucleus. Okay, I'm going to leave that there. I hope you found that useful. Stay tuned, because coming up next, we've got a video on viral structure. And then I think we're also going to post one on HIV replication for those of you that do AQA A-level biology. So switch on those notifications and make sure you are subscribed.